Examples of controls. Oh, I've given you some of these already, but these are written out in the a variety trial. You're using the field variety included inside your trial, hand planted, whatever else. And an experimental test for viruses, you've got the do contain and do not contain. So I already showed you a picture on these two. Oh, actually, I showed you a picture that included all of these, so we'll keep going. Replication. So let's say I have a water and water plus magnesium in my bean plant experiment, and I have the water only has a length of 93 millimeters, but the one that had the magnesium has a length of 108 millimeters. Do I have any confidence in that difference? What if by just chance I was looking at this plant? You see the highest plant with water right here at about 97, 98 millimeters, 97. There's one plant with water that grew 97 millimeters. Here's one plant with water and magnesium that grew about 90. I could conclude if I only had two plants and just by chance it was with water at 97, with water plus magnesium it was 90, I could conclude that magnesium actually reduces shoot length because I didn't replicate. But on the right-hand side, I do replicate, and you can see there's a lot of noise in the experimental units that were measured. Their dependent variable was measured. The dependent variable is shoot length. And you can see all this noise. That noise in the ones with magnesium, all the way from 90 to 114 millimeters, that's the experimental error. Did anybody do anything wrong? Was there a mistake? No. Do not confuse experimental error with an experimental mistake. This is just background noise that you can't control based on genetics of individual seeds, based on how deep was that accidentally planted. Maybe an insect bit one of the shoots as it was just starting to grow and you didn't see it. You couldn't see it. Microscopic damage, but it slowed one of the seeds down just a little bit. There are lots of things that you can't control. That's experimental error. Now, there is experimental mistakes, and you have to be willing to look at that. Did I make a mistake in my experiment? Did I do something wrong? And that's where doing an experiment more than once can help, and also having other labs, other groups redo your experiment, see if they come up to the same conclusions. If they don't, well, why is one lab in Salinas getting a different answer from another lab in Woodland? They should be the same unless you can come up with what's different. What are the two groups doing differently? Maybe one group made a mistake. Maybe one group was working with garbanzo beans of a completely different variety than the garbanzo beans they were using in Woodland. And this two varieties of garbanzo beans just happens to have different responses in magnesium. It could happen. Anyway, I don't want to get too far off on those topics. So this slide explains experimental error. Let's take a look at the cartoon graph we see here, the column charts. And they look like they have little plungers, plungers for, you know, the old-fashioned explosives where you push down the plunger. Well, the plunger is actually an error bar. And the error bar indicates how much variation or experimental error was in the measurements. And so you can see all of them except for one have a relatively small error bar, meaning the range of experimental error, the range of measures of dependent variables in the experiment we're talking about here, the range of shoot lengths in bean plants getting either water or magnesium plus water is relatively small. It's in a relatively modest range. Small experimental error. The one in the middle, treatment C in this cartoon, has a huge error bar. And that huge error bar means we have no confidence in what the real dependent variable value is. What is the treatment effect? What's the real number? It's just all this noise of different numbers. If I were to go back to the, if I were to go back here, you know, I might have numbers all the way from 115 down to 25, all over the place. And a bunch of them high, a bunch of them middle, a bunch of them low. I have no confidence. I have no idea what's going on. Something's wrong when you get that much variation in your dependent variable, your measured variable. Maybe you've got a genetic problem with the seeds you're using. Maybe there could be a number of things, but when you have huge variation, you got a problem. And hence, in this graph, they say, sorry, we just can't trust you. Well, yeah, the data can't be trusted because what is the top of the bar? The top of the bar is the average response for this treatment. Just like this pink bar is the average response in the shoot lengths. So if I were to make a bar graph out of this, I would just box this off as a bar, a column, I should say, of pink, and that would be my two columns from the bar, the horizontal pink bar, all the way down to the x-axis. Now, the error bar only takes what's called the standard deviation. It doesn't take the minimum and maximum. The error bar isn't these two extremes. The error bar is some statistical measure called the standard deviation and would probably be somewhere around this cluster of values. You have the little plunger right about here in this illustration, all right? So let's look at this after that explanation. Let's look at exactly what the slide says. Experimental error is the variation in the responses among the experimental subjects. The response was how tall the bean plant grew, how long the shoot grew. That's the response. The experimental subjects was the individual bean plants. And the variation is where all of those individual yellow circles go, okay? So experimental error is the variation in the responses, or measure, response is the same thing as saying measurement of the dependent variable, same thing, among the experimental units, the individual plants or experimental subjects, which are assigned the same treatment. So in the previous graph, I'm not going to go back again, the previous graph, they separated them out to the same treatment, the ones that did get magnesium and the ones that didn't. So there's something called the analysis of variance. The analysis of variance, AOV, or ANOVA, analysis, AN, of variance, VA. So ANOVA is just easier to say than AOV. And we talk about doing ANOVA in statistics, and we use it like a verb or a noun. It's interchangeable in a way. Oh, I'm going to ANOVA those results. That's not technically accurate, but it can be casually used as a verb, even though it's a type of experimental analysis, a type of mathematical statistics that's done to determine how much variation there is. How much variation really is in those experimental units. So ideally, we would like experimental error to be 0, where our constants, our constant variables were perfect, and our responses of the dependent variables were perfect, but that's not reality. There's going to be at least something going on. There are differences in the experimental subjects. I'm doing a drug trial. I could have only Caucasian males between 50 and 60, period. Forget women, forget ethnic groups and keep them all the same to make the experiment easier to run from a statistic standpoint. And then I'll run different studies on individual groups. I'll do a different study on the women, a different study on different ethnic groups. That also becomes difficult. But no matter how hard I try to make them all the same, can I find 150-year-old white guys that are exactly the same for a drug study? No, you can't. You just can't. And so you have to do your best. There's always variation within the experimental subjects. And even with plants, I don't care how perfect or clonally exact those lettuce seeds are, or those scions from your Chardonnay clone that you're using to graft to that particular rootstock. There are going to be minor differences that you can't change. Differences in the devices that record the measurements. pH meters are not perfect. Even rulers aren't perfect. You're going to have some variation. Sometimes the variation is very tiny and won't change the experiment, but sometimes it can be significant. Variation in applying the settings or treatments. No matter how many seeds I try to plant exactly 1/8 deep, some of them are going to occasionally go a little too deep or a little too shallow. And I add water to the seeds, and I'm going to add a little more water to that pot than the other pot. You can't be perfect. So experimental error is just part of life. There is inherent variation that cannot be controlled. The more money you spend, the more effort you make, the more you can control experimental error to a point, but then all of a sudden your costs start skyrocketing, and the rate at which you continue to decrease experimental error is almost negligible. You're just wasting money. You can't get at the last bit. So control over the magnitude of experimental error, or how much experimental error you have, can be achieved by choosing good experimental subjects. Choose a variety or a clone of vineyard plants that are known to be highly uniform. Choose a block that doesn't have a lot of topography with changes in elevation or slope. So a nice flat block with good uniform soil is much better. Or a really nice south-facing slope that might be sloped some, but all you have to worry about is the top of the slope to the bottom of the slope. You don't have some facing southeast strongly and some others facing strongly southwest around this curve. That really complicates things, because not only do you have strong east-west differences, but you have strong slope, top of the slope, bottom of the slope differences. Stay away from that. That's not a good experimental subject, unless you're stuck with it. Sometimes you're stuck with it. You do what you got to do. Taking care with experimental procedures. So I trained people very carefully to trim heads of lettuce so they were exactly trimmed right, so that we didn't have differences in people trimming. You know, one person trims more aggressively than another person that changes their measurements of mass and core height. You know, making sure that you take good random samples of clusters from your vineyard, etc. Blocking, which we haven't gotten to yet, but when we get to blocking, that will make more sense. And total experimental design. And don't worry about the word covariates. If you have taken statistics before, then using covariates is something that you can do for more complicated situations. But if you haven't taken statistics, you can stick your fingers in your ear when I say the word covariates. So why is replication important? Well, you need to reduce the effect of uncontrolled variation, the experimental error, or controlled variables that cause a change in the dependent variable. Remember, the experimental error is changes or variation in the dependent variable based on unavoidable factors, all right? And you can't possibly have it perfect, so you have to replicate. I've already stressed this. I've given you examples, so I'm not going to say it yet again. But you're trying to put a number on that random background noise, on that experimental error. And the only way you can get a measurement of experimental error is through replication. And the only way you can tell if fertilizer A is better than fertilizer B is if you have a measure of that underlying variation or experimental error. You need replication, or you have nothing that can give you confidence in a result. Randomization: experimental units or subjects under study should be assigned to treatment groups at random. Random does not mean chaos. Random just means that any individual has the same chance of getting any treatment. You don't pick the treatments based on location or age or size or whatever. You don't assign them by some process. It's just random. One needs to explicitly randomize using a computer program, coins, dice, cards, Sharpie labeled pennies, whatever it takes, but make sure it's random. There is a random number generator app. Computers actually, from a really technical standpoint, really have a hard time generating random numbers. It's harder than you think to do. The random number generators on most computers are not truly random, but they're random enough. They're good enough for agricultural experiments. In some areas, like the studies of subparticle physics in the Large Hadron Collider, things where you need just amazing numbers and precision, and randomness in your experimental design has to be perfect. It's really complicated, and I'm not going to get there, but randomization is harder than it sounds. But for agriculture, it's easy because we don't need that level of precision. Ag is kind of a, yeah, it looks pretty good. You do a good job, it's not hard. And Excel's random function, or most any computer random number generator like the one you see in the lower right corner of this slide, they work just fine. Why is it important to randomize? Well, you avoid bias. You don't assign things without even knowing. So you select the beans that look bigger to favor the outcome of a treatment, to control a role of chance. Randomization allows the later use of probability theory, and so gives a solid foundation for statistical analysis. There is chance that comes into play. There's random error, random chance, random occurs in experiments, and by randomizing your treatments, you give yourself a better control over the other random factors. And let me just give you an example of how randomization actually works, then we'll take another short break. I'm going to open up Microsoft Excel, and I'm going to say view, zoom, and I'm going to do 250%, just so you can see it on the screen. And now let's say I've got treatment A, B, C, A, B, C, A, B, C. So those are my treatments. And I want to randomize those against the plots that I'm going to assign. So I've got my plots, or my experimental units. And I'm going to use the random function equals RAND, open parenthesis, close parenthesis. That's it, random function. And now I'm going to copy that down. And now I'm going to copy that function, and I'm going to choose the paste function to paste it as a number so it stops changing. I paste it as a number. So instead of it being the random function, if you look here, it's just now this long number. All right? So all I did was I randomized with the random function, and it creates a random number between 0 and 1. And now I can sort based on column G. And so I use the sort function. Data, sort, and I'm going to sort column G. Now it's randomized. And now I can take these treatments. Oops. Come on. Just so they're next to each other. All right, so now block 9 is going to get treatment A, block 7 is going to get treatment B, block 1 is going to get treatment C, etc. Now I randomize the whole thing together. Sometimes you have to randomize them in groups for blocking. We'll get to blocking later. I didn't want to show you blocking in this at the moment because, well, maybe I should. While I'm here, let me do that. Let me do that. I'm going to do only 2 groups this time, ABC and ABC. I've got 2 different blocks, which you'll understand in a moment, or 2 different groups or replications of my experiment, all right? And I've got plot number 1, 2, 3, and plot number 4, 5, 6. And I have to randomize these separately because they're in separate blocks. Fine, so we'll do that. Equals rand, open parenthesis, close parenthesis, enter. I can just fill down, it's all random anyway. Notice every time I change this, it keeps changing the numbers. So I'm not worried about it, it's still random. And now I'm going to copy. And I'm going to choose paste. Paste special as values. And now I've pasted the numbers. Now they won't change. So I'm going to sort this first group. Data, sort the first group. And I'm going to sort the second group. Data, sort G. So I've sorted them from the lowest to the highest value that was randomly generated. So in this group, treatment A is going to go in the first block or first group of plants. Treatment B is going to go into the third one, and treatment C is going to go into the second one. In the second block, the second group of plants, treatment A is going to go into the fourth block, or fourth plot, I should say. B is going to be the fifth block, C is the sixth block. It just so happens that they're in order. That's just a random chance. With a number as small as 3 things, it's not too unlikely. All right? So that, my friends, is how I randomized with Excel, and I could then turn that into a very simple field layout with my plots. This is the plots for my field map or my experimental subjects and my treatment. Fertilizer 1, fertilizer 2, fertilizer 3, whatever. All right, so let's take another break. I know this is a long presentation.